If we were playing a word association game right now and I just said the word mitochondria, I know exactly what you would scream back at me.
Powerhouse of the cell.
Exactly. I mean, it is arguably the most successful branding campaign in the history of biology.
Yeah, it really is.
You learn it in middle school and it just sticks forever. But there is a problem with that phrase. Because when I think of a powerhouse or a battery, I picture this buzzing, static box in the corner of a room. It just sits there, humming, doing one thing.
Which is a massive disservice to what is actually happening inside your cells.
Right.
Because if you could shrink down and actually stand inside a mitochondrion, it wouldn't look like a static box at all. It would look like Tokyo or New York City during rush hour.
Wow.
It is a bustling, chaotic, just incredibly complex metropolis.
And that is exactly what we are unpacking today for you. We aren't looking at the energy output of the mitochondria. We are looking at the citizens of this city. We are looking at the social network. We're taking a deep dive into a study published in Nature called Mitochondrial Complexome, reveals quality control pathways of protein import.
It's a bit of a mouthful of a title, but it is a landmark paper.
It really is. And the reason we picked this one for today's deep dive is because it solves a problem I honestly didn't even realize we had. I just always assumed scientists knew how the mitochondria worked. Like, we have the parts list, right?
We do. We have what's called the proteome. So we know the names of the thousands of proteins, the workers essentially, that live inside the mitochondria.
Okay.
But knowing the names isn't the same as knowing the jobs, or more importantly, knowing the teams.
It's kind of like having a directory of every employee at a massive corporation. Let's say Google.
Yeah, good example.
You have the list, but you have zero idea who works in marketing, who works in engineering, or, who is meeting in the conference room on the third floor.
Exactly. We have the employee list, but we didn't have the org chart. We didn't know the interactome, who hangs out with whom to actually get the work done.
Right.
And without that wiring diagram, you can't really understand the machine. And you certainly can't fix it when it breaks.
So the mission of this deep dive is to look at how these researchers finally built that org chart. They created something they call MitCOM. And in the process, they didn't just draw a map. They found a secret security team hiding in plain sight that nobody even knew existed.
A security team that literally decides whether the cellular city survives or collapses.
Right.
But before we get to the high-stakes drama at the gates, we really have to talk about how they built this map. Because the technology here is honestly just as cool as the biology.
Yeah, let's get into the MitCOM tech. Why was this so hard to do before? I mean, why couldn't we just look under a microscope and see who was holding hands?
Well, because proteins are incredibly tiny, and their interactions are very fleeting. Traditionally, if you want to identify a protein, you have to run it through a mass spectrometer.
Okay.
But to do that, you usually have to denature it.
And denature means unravel, right?
Right. You take this complex 3D folded knot of a protein and you stretch it out into a straight line to read its code.
Gotcha.
But the moment you unravel it, you lose the connection to whoever it's holding on to. You break the team apart to identify the individual players.
So you know who was in the building, but you completely lost the information about who they were sitting next to.
Precisely. So this study used a technique called CSBNMS, which stands for complexome profiling, using blue native electrophoresis and mass spectrometry.
Okay, that is an absolute alphabet soup. Let's strip the jargon out. How does it actually work in practice?
Think of it like a foot race. You take all the proteins out of the mitochondria, but you keep them in their native state. You don't unravel them.
So you leave the teams intact.
Yes. Then you put them in a gel lane and apply an electric current.
And because of physics, the heavy things move slower than the light things.
Exactly. The massive supercomplexes, think of them like full ropey teams. They get stuck near the starting line at the top of the gel.
While the tiny solo runners sprint to the bottom.
Right. So you end up with a gradient of teams sorted entirely by size.
Okay, so you have this gel with the heavy stuff at the top and light stuff at the bottom. But here is the twist that I loved when I was reading the source material. Usually, scientists would just chop that gel into a few big chunks to analyze it. But these guys went a little crazy with the slicing.
They really did. They used a cryo slicer to cut that single gel lane into 245 tiny distinct pieces.
245 slices.
Yeah, and each slice was only 0.3 millimeters wide.
Yeah.
That is significantly thinner than a fingernail clipping.
And why go to that extreme? I mean, is it just showing off at that point?
No, it's all about resolution. By slicing it that thin, they could pinpoint exactly where a protein was peaking in the race.
Okay.
If they found protein A peaking in slice 50 and protein B also peaking in slice 50, and their abundance profiles matched perfectly in slices 49 and 51.
Then it's not a coincidence. They are physically stuck together. They are in a complex.
Correct. We call that co-migration. And they did this for over 900 distinct mitochondrial proteins. It generated this massive data set.
Wow.
And when they started looking at that data, the first thing they found was a statistic that honestly completely changes how I view biology.
I think I know which number you're going to bring up. The 6.4.
That's the one. They found that the average mitochondrial protein isn't just found in one spot in the gel. On average, each protein appeared in 6.4 different peaks.
Let's just pause and let that sink in for a second. In our city analogy, or the corporate directory analogy, that means the average employee doesn't just have one job, they have 6.
Basically, yes. We used to think of proteins as highly specialized, like, I am a respiratory enzyme, my only job is respiration.
Right.
But this data suggests that the cellular economy is actually a gig economy. Most proteins are moonlighting.
So the guy who works in accounting is also the backup goalie for the soccer team, and he drives for Uber on the weekends, and he volunteers at the fire department.
Exactly. A protein might be building energy in one complex, but in another assembly, it might be stabilizing a membrane wall or acting as a chaperone for a VIP protein.
That is wild, because it implies that our current understanding of biology, where we tend to assign one function to one protein, is probably missing like 80% of what is actually happening.
Is a very humbling realization.
Mm-hmm.
We're looking at their day jobs, completely unaware they are working the night shift at the emergency room.
Speaking of the emergency room, let's zoom in on the specific discoveries. Once they had this MitCOM, they started seeing connections that resolved old mysteries.
They did?
And one of the biggest ones involves the entry gate.
Right. So the mitochondria is a fortress. It has two membranes, an outer wall and an inner wall. And it has to import thousands of proteins from the outside to function properly.
And to get in, those new proteins have to pass through a specific gate called TOM, right?
Yes. The translocase of the outer membrane. Good old Tom.
I just love that the molecular bouncer is named Tom.
Friendly name. Very tough job. Now, we've known for a while that ribosomes, those are the factories that build proteins, they hang out near the mitochondria.
Okay.
But we weren't sure exactly how the handoff happened. Does the ribosome just dump the new protein near the mitochondria and hope it floats into the gate?
Like an Amazon driver just tossing the package vaguely toward your port.
Exactly. Or is it a precise handoff? MitCOM gave us the answer. They found a protein called UM14.
And what does UM14 do?
We knew UM14 acts as a receptor for ribosomes, but the map showed UM14 physically docking with the TOM complex.
So it's not tossing the package on the lawn, it's backing the delivery truck all the way up to the loading dock.
It's a direct physical link. A ribosome docks onto UM14, which connects right to TOM, and a new protein is pushed through the gate as it is being built. We call it co-translational import.
That makes perfect sense. It's super efficient. But this brings us to the central drama of the whole paper.
Oh, yeah.
Because if you have thousands of proteins trying to squeeze through one main gate, eventually something is going to go wrong.
Traffic jams. That is the crisis this paper focuses on. When a protein gets stuck in the TOM channel, it is a disaster for the cell.
Like a semi-truck jackknifing in a tunnel.
Exactly. Like in the Lincoln Tunnel.
Yeah.
Nothing goes in. If you don't clear it, the mitochondria starves, the energy stops, and the cell dies.
So how do they clear the tunnel? This is where the researchers found what looked like a glitch in the matrix. They saw a protein at the gate that had absolutely no business being there.
They were looking at the TOM complex on their map, and they noticed a protein called PITH2 co-migrating with it.
PITH2. And why was that weird?
Because PITH2 is a peptidyl tRNA hydrolase.
Okay, translation please. What does that actually mean?
In plain English, PITH2 is a housekeeping enzyme. It usually just floats around in the fluid of the cell, clearing out waste products from protein synthesis, specifically stuck tRNA molecules.
Okay.
It's essentially a janitor for free-floating trash. It isn't a membrane protein. It isn't a gatekeeper. It had no historical business being attached to the main security gate of the mitochondria.
So it's like finding the guy who sweeps the break room, suddenly checking IDs at the high-security entrance to the Pentagon.
That is exactly what it's like. The researchers were skeptical. They thought, is this an artifact? Is our map wrong? So they ran a test.
What kind of test?
They physically fished out the TOM complex from the cell using affinity purification to see what actually stuck to it in real life. And sure enough, PITH2 was holding on tight.
So he really is at the gate. But is he doing anything? Or is he just loitering?
That's the next logical question. So they deleted the PTH2 gene entirely from the yeast cells to see what would happen. We call this the break it and see what smokes method of biology.
Which is usually the most effective method, honestly. So they fire the janitor. What happens to the building?
Chaos. Without PEEth2, the system started to fail. Precursor proteins, the ones trying to get inside, like one called MDJ1, started piling up outside the gate. They couldn't get in.
So the janitor was actually critical to security.
PEEth2 wasn't just hanging out. It turns out it's part of a coordinated response team, specifically designed for when the gate gets jammed.
Wow.
But here's the thing. PEEth2 doesn't work alone. This is where the story gets really elegant.
Yeah.
When a protein gets stuck in the gate, the cell doesn't just yank it out immediately. It tags it first.
This is the ubiquitin system, right? The cellular trash me sticker.
That's the one. Ubiquitin is a small molecule that cells attach to proteins to signal, this is trash, process this. The researchers looked at their MitCOM map to see who was holding the staple gun near the gate, who was putting the tags on the stuck truck.
And they found a protein called Rsp5.
Right. Rsp5 is a ubiquitin ligase. The map showed it hovering right at the tom gate. Its only job there is to slap that ubiquitin tag on the protein that's jammed to the door.
Okay, so step one, tag the wreck. But then they found another protein, UBP16, and this is where I got genuinely confused reading the paper.
That throws a lot of people.
Because UBP16 is a ubiquitylase, meaning it removes tags.
It does.
That seems incredibly counterproductive. You have Rsp5 putting stickers on and UBP16 ripping them off. Is this just bureaucratic nonsense at the cellular level?
It really does sound like it at first glance. But think about the physics of the situation. You have a protein stuck in a tiny pore. You've just covered it in sticky ubiquitin tags.
Right.
Sometimes those tags get messy. They form long chains, or they branch out in weird directions.
Like a tangled ball of tape?
Exactly. If the tag is too messy, it might actually make the protein harder to pull out of the gate, or harder to feed into the shredder later on.
Oh, I see. So UBP16 isn't removing the tag to save the protein, it's grooming the tag.
Precisely. It's trimming the label so the barcode can actually be read. It modifies the ubiquitin chain so that the extraction machinery can get a good, clean grip on it. It's quality control for the trash signal itself.
Okay, that makes so much sense now. So we have the tagger, which is Rsp5, and the groomer, which is UBP16. But we still have a wrecked truck in the tunnel. Who brings the tow truck?
That brings us back to our mystery guest, PC2.
The break room sweeper turned security chief.
It turns out PEEth2 is the dispatcher. It sits at the gate. It recognizes the situation. Okay, we have a clog, it's been tagged, it's been groomed. PEEth2 then physically recruits a protein called DSK2.
And DSK2 is the tow truck.
DSK2 is the shuttle protein. It has one spot that locks onto PEEth2 and another spot that grabs the tag trash. It physically hauls the stuck protein away to the proteasome, which is the cell's incinerator.
So PEEth2 is the critical link. It connects the problem at the gate to the solution in the incinerator.
And that connection is absolutely vital. When they genetically removed DSK2, the tow truck, the clogs piled up just as bad as when they removed PEEth2.
So they depend on each other.
Right. They're part of the exact same linear chain of command. Rsp5 tags, UBP16 refines, PEEth2 calls the truck, and DSK2 hauls it away.
It's a dedicated waste management workflow just for this one specific type of traffic accident. That is incredibly cool. But I have to play devil's advocate here for a second.
Go for it.
This seems like a lot of highly specific steps for a single cell. And biology usually hates having a single point of failure. Is this the only way to clear a clog? What if PEEth2 is on a coffee break?
That is a great question, and it's one the reviewers of the paper almost certainly asked the researchers. Biology loves redundancy. If you only have one safety valve and it fails, the organism dies.
Right.
We actually already knew about another pathway called mitotad.
Okay, so if we have mitotad, why do we need PEEth2 at all?
That's what they checked. Are these systems just backups for each other, or do they work together? And they found that they are completely independent.
Interesting.
If you knock out PEEth2, the mitotad system, which involves proteins called UBX2 and CDC48, keeps working just fine. But it's not enough to handle the load alone. They act as separate, independent safety nets.
So maybe they handle different types of clogs or work under different stress levels.
Exactly. Or maybe one handles clogs that happen deep inside the gate and the other handles clogs right on the surface. The study also mentioned that Preth2 is linked to a system called VMS1, which helps clean up stalled ribosomes.
Wait, the ribosomes from the loading dock we talked about earlier?
Yes, so Preth2 is really a guardian of the gate in a very broad sense. It's watching for stuck proteins and broken factories. It's a multi-talented crisis manager.
Which brings us back to the big picture here. We started this conversation talking about how we usually visualize mitochondria as simple batteries. This study just completely shatters that, doesn't it?
It's a complete shift in perspective. We've gone from the mitochondria makes energy to the mitochondria is a self-regulating city with traffic control, emergency response teams, and a highly specific waste management infrastructure.
And I guess the question for you listening right now is, why should you care about yeast traffic jams? I mean, it's fascinating science, but does it actually matter to us?
It matters immensely because mitochondrial traffic jams happen in human cells too, and when they happen, neurons die.
Oh, wow. So this isn't just a yeast problem.
No, not at all. Failures in mitochondrial quality control are a hallmark of some of our most devastating diseases. Parkinson's, Alzheimer's, many metabolic and cardiovascular disorders. They all involve the failure of the cell to clean up its own trash.
So if the tow truck system fails in your brain cells, the waste piles up, the power plant shuts down, and the cell dies.
Exactly. By mapping these pathways in yeast, by finding Peth2 and Rsp5 and UBC16, we identify potential targets for medicine.
Because now we know what to look for.
Right. If we know the machine is broken because the tow truck isn't being called, maybe we can design a drug that mimics that call, or a drug that boosts the activity of the groomer. You can't fix the engine if you don't know where the spark plugs are.
And MitCOM just gave us the schematic.
It did. It gave us a Google Earth view of the mitochondria. We could zoom all the way in from massive supercomplexes down to specific security guards. It gave us the wiring diagram we've been missing.
That's the part that really sticks with me. I keep thinking about that 6.4 number we started with. The idea that every protein has 6 different jobs.
It keeps me up at night, honestly.
I mean, we just spent all this time unpacking one new role for one protein, Peth2. If every single protein in that list of 900 has 5 or 6 other jobs we don't know about, are we looking at a revolution in biology?
I think so. We are moving away from reductionism, studying one thing in a test tube, to complexomics, studying the whole messy, interconnected network. We just have to assume that everything is doing more than we think it is.
It's daunting, but really exciting. It means there are still plenty of continents left to map inside the cell.
Absolutely. We've really just scratched the surface of the slice.
Nice callback to the gel slices.
Hmm.
Well, that's all the time we have for this deep dive into the mitochondrial city. Next time you feel tired, just remember, you have trillions of tiny security guards working overtime to keep your gates clear.
Hopefully Peth2 is on the job.
Thanks for joining us on this deep dive. We'll catch you on the next one.